So today we'll talk about a paper that deals with the ERK pathway. The paper says activation of the RAS-ERK signaling and JSK3 by amyloid precursor protein and amyloid beta facilitates neurodegeneration in Alzheimer's disease. It was published some years ago by a group out of UC Irvine. And essentially, in the introduction, they tell us that while when the paper was published, which was, I think, 2017, the contribution of the amyloid beta oligomers and peptides to Alzheimer's disease pathology is or was well understood, little is known about the physiological function of the APP holoprotein or its role in AD pathogenesis. And it's true, like everybody is generally concerned about APP simply as a source of amyloid beta peptides and oligomers. And that's true. Sometimes there are things that turn very toxic in our bodies and in organisms in general, and their source is something that is really good, right? And we start getting concerned as to how come something good produces something so toxic, and that's when we start going down roads like BACE1 and gamma secretase and what is that cleaving APP into these really toxic molecules and this and that. These guys are saying, let's take a step back and let's ask about APP itself, if it might be contributing to progression of the disease by itself in a way that makes it not such a good thing anymore, right? So that's the overall message of the paper. Let's look at how APP may contributing to progression of Alzheimer's disease. The authors then introduce us to the idea that kinases such as JSK3, JNK, and then this guy who's the main character in today's story, the extracellular signal regulated kinase or ERK, and others, CDK5 and 4, CDC2, phosphorylate APP in threonine 668, and this in turn enhances or facilitates the proteolysis of APP by beta secretase, which in turn generates the amyloid beta peptides. And I'm thinking, is that a typo? Is it beta secretase or gamma secretase? Anyway, the secretase that will cleave APP in a way that it generates the toxic amyloid beta peptides, right? So we knew that, and we had mentioned it in class before, that phosphorylation of APP at this crucial site bends the protein in such manner that it makes it more prone to be cleaved by the peptidases that will generate the toxic forms. And perhaps not surprisingly, Alzheimer's brains or Alzheimer's disease brains show activation of these kinases and increased levels of phosphorylated APP, right? But so far, it's all very easily explained by the fact that, sure, there is this upregulation of these kinases, more phosphorylation of APP, makes it easier to cleave APP in a manner that generates the toxic forms, hence you get Alzheimer's disease, right? But what the author says, hold on a second, because we showed in previous work that if you take culture neurons and you put a lot of APP in them, you can enhance RAS expression and activation of ERK1 and 2. We'll discuss that pathway in more detail in a few slides. But for now, the idea is that they took neurons, they put a lot of APP in them, and now there was activation of this pathway. And what they hypothesized is that APP itself, regardless of it being phosphorylated and being cleaved in a way that generates the toxic forms, by itself, it could promote RAS-ERK signaling and enhance proliferation in cells, right? So far, we're not saying anything that ties it back to Alzheimer's disease. We're saying that APP could induce proliferation in cells. But at the end of the paper, they bring it all together and they sort of tie back to the progression of Alzheimer's disease. So what they're saying is APP is not just sitting there doing good things, and at times it can cleave in a way that it generates toxic peptides. It might have something to do with driving the disease itself, and that's what this paper is about. So the very first thing they do is to corroborate some observations that had been made before, and that is they took post-mortem samples of people who never had Alzheimer's disease. Those would be the NAD controls, no Alzheimer's disease. People who, by the time of their passing, had been diagnosed with mild cognitive impairment. So these are individuals who were thought to be experiencing early Alzheimer's or late Alzheimer's disease. These are individuals that were showing many of the more aggressive forms of cognitive impairment and dementia. And they took those post-mortem samples, isolated proteins from them, and they measured a whole bunch of things. So they first looked at APP itself and its proteolytic derivatives, and they see, for instance, that as the disease progresses, so going from left to right, the amount of full-length APP decreases, and there is a concomitant increase in the amount of amyloid beta peptides, right? So as these brains are experiencing disease progression, more and more of that APP is being cleaved into the toxic forms. We knew that, right? So amyloid beta peptides accumulate with AD progression. We knew that. They also look at these samples using an antibody, PHF1, that is designed to detect phosphorylated tau, but that is known to be particularly reactive against neurofibrillary tangles, right? So when these samples will have in them some of these NFTs that accumulate in neurons of very late disease patients. And sure and behold, or long and behold, there is this accumulation of the NFTs in late Alzheimer's disease. They didn't detect any in early Alzheimer's disease or obviously in controls. So, like I said, we knew this before. As disease progresses, there is accumulation of A-beta peptides and oligos, and there is accumulation of NFTs. Then they look at the phosphorylation of these two things, of tau and its contribution to the formation of the NFTs, and of APP itself, right? And what they see is that the phosphorylated tau that will form the neurofibrillary tangles, now they're looking at phosphorylation specifically at threonine 231. So this is the phosphorylated form of tau that is the one that will trigger the formation of NFTs. And sure, somewhat as expected, there is a lot of accumulation of phosphorylated tau in late Alzheimer's patients, not so much early Alzheimer's or controls. With APP, the situation was a bit more interesting. They start detecting phosphorylation of APP earlier on in the progression of the disease. So in MCI patients, they can already see some phosphorylated APP. And of course, phosphorylated APP is present in late Alzheimer's as well. So what this was telling them is that APP and tau get increasingly phosphorylated as AD progresses, and we kind of knew that. The one thing that was a little more novel about this experiment was the phosphorylation of APP itself. The tau stuff everybody knew by this paper, but the phosphorylation of APP was somewhat new. So we can wake up and get your fluid, juices flowing. What can cause the accumulation of phosphorylated APP and phosphorylated tau? What mechanisms can you guys think of that will cause an increase in the levels of these two things? What's Isn't
that phosphorylation maybe prevents them from being cleared from the cells or recognized by anything that degrades them or breaks them down?
Perfect. So I'm going to write that down as reduced clearing or degradation. Love it. So reduced disposal, right? Increased kinases. So it's not so much that these things are not being disposed of, it's just that there is more of them being produced.
I think it was more of like pointing in the right direction. I think like when AD progresses,
Mm-hmm.
these plaques build up, it causes like a cascade within like the system itself that causes the hyperphosphorylation of these two proteins?
So there is hyperphosphorylation, right? And that would explain their accumulation, the increase in their amounts, right? But mechanistically, that's what I was asking. So that yes, there is. You could have an increase in the kinases that will phosphorylate these proteins. So there is the same amount in a way of APP and the same amount of tau. It's just that now you have more kinases phosphorylating them. You're bringing in that snowballing, that vicious circle of more and more and more, and I totally agree. But mechanistically, there could be more kinases phosphorylating these things. There could be reduced mechanisms of getting rid of them. Ale, you had a comment?
I was gonna add, I guess, to increase kindness. Maybe there's not like a physical, like more amount of kindness, but just its activities being increased.
Beautiful. So not so much an increase in the number of kinases present, phosphorylating these things. Your words, it's just the name, the same number of kinases, it's just that they're more active now, right? So activation of the kinase. So I'm going to say increase kinase levels or activity. Yeah, Josh.
Okay, how about a little bit more specific? An earlier article said that AB would damage the DNA. Would the damage the DNA actually affect some of these processes? Like mechanistically?
Well, so that is mechanistic in a way. It's just that is placing that mechanistic step a bit earlier. You're simply saying the presence of these AB oligos at low amounts will kickstart some DNA damage. We did discuss that. And then it was the DNA damage that, I don't know if you guys remember, but there was inflammation in between, and that inflammatory process led to more and more accumulation. But at the end of the day, what is that will give you more phosphorylated tau and more phosphorylated IP? And yeah, we continue going around some of the same arguments. More kinases or the same kinases more active. Also, you mentioned something that prevents the clearing of these things, and I totally agree. What other ways, aside from getting rid of the whole thing, could you do to reduce the levels of phosphorylated tau and phosphorylated APP? Yeah. Phosphatases. So that's sort of the counterpart of increased kinase levels or activity is the reduced phosphatase levels or activity of any phosphatases that would come and dephosphorylate these things, right? So the counterpart of increased phosphatases is reduced. Sorry, the counterpart of increased kinases. I'm going to say levels or activity. And then there is one that we tend to forget about. Marcia? The amyloid beta, what do you mean? The Sure, but in a way, like Josh was saying, you're asking the question or addressing the question, what triggers this? We're essentially trying to understand what is it that researchers typically go for once they see an accumulation of phosphorylated APP and phosphorylated tau. Mechanistically, what are the things that they might look for next, right? So we know that there is more phosphorylated APP and phosphorylated tau. What is that could be going wrong? And we already heard ideas about increased levels in the kinases that normally phosphorylate them or their activity, reduced levels of the phosphatases that normally dephosphorylate them or their activity. And then there was this idea that they accumulate simply because whatever mechanism is normally clearing them or degrading them, those were the right words, are no longer operational, right? But there is one other thing that we tend to forget. How else could you increase the levels of phosphorylated APP and phosphorylated tau? If what gives you phosphorylated APP and phosphorylated tau is taking an APP molecule or a tau molecule and phosphorylating it, what else could give you a lot more phosphorylated APP and tau? Oh, increasing the production of ATP, okay. Same thing. How about simpler? So not wrong, but how about simpler? What else could you increase that will give you more phosphorylated APP and phosphorylated tau? See, this is beautiful because we tend to forget this, or we tend to skip this. Fats, again, so you're going back in the chain of causal events. Yeah, one last time.
Phosphates in cells?
Well, so that's like ATP. So we're gonna stop right here. But it's beautiful because it shows what is a very common instinct in all of us. We immediately think, oh, if I see more phosphorylated APP and I see more phosphorylated tau, it has to be that there is the kinases, the phosphatases, the ATP, the fats, the proteins themselves, right? APP and tau themselves. Remember that anytime that in cells we see an increase in the amount of activated or inhibited something, yes, it's okay to look at the mechanisms that activate them or inhibit them outside of themselves. But then there is the levels of themselves too. So something that increases the amount of APP expression or of tau expression, assuming same levels of everything else, same levels of kinases, same levels of kinase activity, same levels of phosphatases, same levels of phosphatase activity, same ATP levels, same fat, same amyloid beta. Everything else being equal, if you raise the baseline amount of APP and the baseline amount of tau, you could have more phosphorylated forms of these two. Okay? That is why these people are, anytime that they look at, and we've seen this before, anytime that they look at phosphorylated tau, for instance, they will look at total tau as well, just to make sure that the increase, or the accumulation of phosphorylated tau is not easily explained by an accumulation of tau, total tau itself. When they look at phosphorylated APP, they also look at full-length APP, just to make sure that that accumulation of phosphorylated APP is not easily explained by an accumulation of the unphosphorylated forms. Okay, so with all of that said, and since in the previous plots they had seen that total levels were unchanged for APP and tau, they decided to start exploring some of these other ideas that you guys had, like activation of the kinases that phosphorylate them, right? And in the introductory slide, we heard that when it comes to tau, for instance, there's a bunch of, and APP as well, a bunch of kinases that can phosphorylate APP and tau. The ERK kinase and the GSK kinase being two of them. Right? So they decide to look at the activation levels of these two as disease progresses. And I will have you first look at the total levels of the enzymes themselves, shown in the ERK and the GSK blots. And sure, there is some variation across samples. These are post-mortem samples, so these were different individuals that may have had different amounts of these things in their brains. But then they also looked at the phosphorylated forms of ERK and GSK, and they noticed that when it comes to ERK, there isn't a super tight correlation between disease progression and phosphorylation of ERK. You can see that the levels of phosphorylation of ERK are somewhat comparable between early disease and late disease. So there may be a correlation with disease overall, but it's not super tight in terms of disease degree, right? Or intensity. And with GSK, it was sort of the opposite. As disease progressed, there was less and less and less phosphorylation of GSK. Yet, the authors conclude that ERK, and I have it in parentheses there because that correlation is mild, and GSK3 get progressively activated with Alzheimer's disease. And this is, they claim, relatively new in terms of knowledge and information. No one had seen this before. So now we have phosphorylation of ERK being somewhat constant, higher overall between disease patients, but you have some normal patients with pretty high levels of phospho-ERK, so That's a mild correlation. And then we have GSK phosphorylation clearly going down, and yet they say that GSK is getting activated. So my question to you guys is, how can phosphorylated ERK turn it on and activate it? And phosphorylation of, I kind of misworded the question, but how come that we're saying that GSK3 is getting activated even though its phosphorylation is less and less over time? In other words, why would phosphorylation of ERK help with ERK's activity, but phosphorylation of GSK inhibit its activity, and therefore you need to lose the phosphorylation in order for the kinase to become active? Activated somehow. So we have that, what we're saying is that in one enzyme's case, the phosphorylation somehow activates it, and in the other enzyme's case, the phosphorylation somehow turns it off, right? And Jessica, you said maybe it blocks something. What could it block that matters to that enzyme? The active site, for instance. So the phosphorylation of the enzyme could somehow block the active site of GSK3 such that now the real substrates cannot access that active site and be phosphorylated by GSK3. If that were the case, then we would imagine that phosphorylated GSK3 is an inactive GSK3, and only when it gets dephosphorylated can it start doing its job. So I totally agree, phosphorylation could block an active site. A more general question, even if it's not physically blocking the active site of the enzyme, how can a phosphorylation event alter the activity of an enzyme, a kinase or any other enzyme?
Phosphates are negatively charged, so could it prevent like interactions via charges, like ion charges?
Beautiful. And I'm going to sort of slightly edit that answer. It's not that it prevents the interaction, it's just that it alters them, right? So a phosphorylation event means ultimately introducing a negative charge into a protein, a polypeptide. And by introducing that negative charge, the rest of the polypeptide will need to accommodate to that new situation. So those intramolecular interactions are altered such that now the whole protein will respond with what we call a conformational change. You guys may have heard this many times before. And that is what, by changing the shape of the enzyme, we can change the activity of that enzyme. And this happens to kinases, phosphatases, all kinds of enzymes in cells.
Well, why wouldn't you fully inhibit it? Like, you're saying it would alter, it wouldn't...
What I'm saying is that it alters the shape of the enzyme, and by altering its shape, it's fully inhibiting it.
Yeah. Okay.
Right? So it's not that it's altering its activity, it's turning off its activity by altering its shape. Right? By a way of hands, who has heard before of this idea of structure equals function? Right? Yeah, that's someone asked me many years ago, how would you define molecular biology? And I didn't invent this, but I'm just a messenger. But molecular biology can simply be defined as that, as understanding that connection between structure and function, right? So when it comes to enzymes, we go around saying that enzymes do what they do because they have a certain structure. They have a certain shape in space that allows them to catalyze reactions that would otherwise not happen within cells, right? That's intro to biochemistry stuff. What we tend to forget is that anything that has to do with the dynamics of biology and regulatory processes and all these functional crosstalks and whatnot, it always goes through altering the shape of things. When we say this thing gets phosphorylated, dephosphorylated, proteolytically cleaved and whatnot, it always comes down to we're changing the shape of these things in space, and therefore by changing their structure, we're changing their function. The only thing that we need to be super careful about is we're changing in the case of an enzyme, for instance, in most cases we're changing the tertiary structure of the enzyme. The primary structure, that sequence or that chain of amino acids, we're not altering, right? So we're not changing the sequence of amino acids in the protein, but we're changing the shape that chain of amino acids adopts in space, because now one of those amino acids has a phosphate in it with a negative charge that is making it fold differently. Yeah? Questions or comments, anyone? All right, and then I have another question for you guys, and that is, fine, all of this is correlations. We see disease progressing in these samples, and we see that certain things are going up, certain things are going down. So if we wanted to prop causation, we need to drive AD in a controlled manner, meaning now instead of looking at what nature did for these individuals before they passed, we need to take the reins and say, now I'm going to drive AD progression in an experimental system, and so that I can start asking questions of what comes first, what comes afterwards, what can I block as an intermediate and see what disappears and what stays, and all these things that we normally do in genetics, right? So my question to you is, what is one of the main drivers of AD that we've been discussing over and over? What's one of these things that you can do to nervous systems and they will almost inevitably go down the path of AD progression? That's right. The A-beta oligos, paper after paper, we see that any of the various aspects of AD progression, they kind of always start with this accumulation of A-beta, either the peptides themselves, particularly in their toxic form, the oligos, right? By the time that we get to the plaques, I think now we all know it's a bit too late now, right? That's one of the late steps or late stages, right? Cool. So yes. So what can we do to a brain so that it slowly develops AD? Incubate it with A-beta peptides. And this is what the authors did. They took neurons in culture and they incubated them in the presence of DMSO, that's simply an organic solvent that was the vehicle for dissolving the A-beta peptides. A-beta peptides, given their hydrophobic nature, they won't easily dissolve in water, so you need some sort of organic solvent, and DMSO is a pretty common one. So when in a paper you see DMSO or ethanol, that's simply the authors telling you this was the solvent that we use for the drug, but in this case we're using the solvent alone without drugs, so they use that as a control for what the absence of drug is doing to your cells, right? So in this case, cells treated with DMSO is almost like saying, I haven't really treated these cells yet. And when they treat the cells with DMSO, obviously you're not going to see any AB oligomers in, or I suppose not detectable levels of them because you haven't added any yet. The total levels of ERK and phosphorylation levels of ERK are what they are, baseline, and then actin was used as a loading control. When they did add A-beta oligomers at 2 different concentrations, kind of hard to tell based on the blood images, but there was an accumulation of phosphorylated ERK, even though the levels of total ERK didn't change much. And when something is hard to see by eye, people will do these densitometries to let computers figure out how much there really was of things. And when they did that, they calculated a relative density of phosphorylated ERK to ERK, and they see that the DMSO, it was that baseline level, and with incubation of A-beta, you see a significant increase in phosphorylated ERK. Okay? So now is a little less of a correlation. Now, I'm adding the A-beta, and I'm causing an accumulation of ERK. All the previous blots that were showing that loose correlation between phosphorylated ERK and Alzheimer's, someone could have argued that phosphorylated ERK was causing Alzheimer's, right? These guys are turning around and saying, well, no, I trigger Alzheimer's with A-beta, and I see this accumulation of phosphorylated ERK. I said that I would introduce the pathway. I said I would introduce the pathway, the ERK pathway before. So stole this cartoon from a cartoon that the authors have in the paper. You'll recognize this cartoon later on. I kind of edited it so that we isolate the ERK pathway. In general, the ERK pathway is initiated by a whole bunch of different receptors that sit on the membrane. And these receptors, sometimes they have the capacity to phosphorylate things inside of cells themselves. Sometimes these receptors pair up or partner with kinases that are sitting in the membrane, and they phosphorylate things that are in cells. In this case, very generic cartoon, you have your receptor on the membrane, it detects the presence of some ligand, and that somehow leads to phosphorylation events within the receptor molecule itself, right? This is very generic stuff. This is how ERK activation happens across many different signaling systems. That phosphorylation event of the receptor molecule near the membrane, what that does is it brings this molecule, this adapter molecule called GRAP2 for short. It's got an acronym, but I don't remember it now. So GRAP2 I should say, is floating around somewhere in the cytoplasm, but now when there is these phosphate groups in certain receptor molecules, GRAP2 loves those phosphate groups in receptor molecules and will bind to them. So now GRAP2 effectively is recruited to the cell membrane. Before it was floating everywhere, now it's all at the cell membrane. GRAP2 in turn will bring this molecule called SOS. And again, SOS was floating around everywhere, but now it's accumulating at the cell membrane. And the reason why it matters that GRAP2 and SOS accumulate at the cell membrane is that one very important molecule that lives on the cell membrane is RAS. RAS is a molecule that can exist in two forms. It can exist in a form that is bound to the diphosphate nucleotide GDP, or can exist in a form bound to the triphosphate nucleotide GTP. Right? So it can bind to either. When it's bound to GDP, it doesn't really do anything. It's inactive. But when it binds to GTP, RAS becomes activated. Right? And when RAS becomes activated, what it will do is it will interact, and by ways of altering the conformation of its partner RAF, it will activate RAF. And RAF is a kinase. So RAF will initiate one of these kinase cascade events by which RAF phosphorylates and activates MEK. MEK is another kinase that phosphorylates and activates ERK. Right? So there is this cascade of phosphorylations in which one kinase phosphorylates and activates the next, and again and again and again. When ERK gets phosphorylated, it will go inside of the nucleus, and inside of the nucleus, it will start phosphorylating some transcription factors, change their shape, allow them to bind DNA. And now these transcription factors that were before unable to bind DNA become able to bind DNA and trigger the expression of target genes. So this whole thing is a way to link the presence or absence of certain molecules outside of the cell to changes in gene expression within that cell's nucleus. All of these guys are always in the cell. The question is whether they're active or they're inside of the cell in their resting form, in their inactive forms. So whenever we see that across those samples, for instance, the levels of ERK were pretty much steady across the board, what that means is that ERK, the protein, was there at a given level and didn't change much from sample to sample. But then you started seeing more and more of the phosphorylated ERK. All that means is that more and more of these ERK molecules, which, once again, were always there, have become phosphorylated. And based on what we know about the pathway, have become active. Okay? So it's like saying ERK is there all the time, but only when you turn it on through phosphorylation will you get that phosphorylated ERK turning on gene expression. The same goes for GRAP2, SOS, and RAS. They're always there in the cell. It's just that they're in different parts of the cell. They're not interacting with one another. For GRAB, SOS, and RAS to interact with each other, what you need to do is concentrate them physically in one place. And that is what these receptors do. They accumulate GRAB2 near the membrane. Now, if an SOS molecule bumps into a GRAB2, it will stay near the membrane, and together they will be way more likely to find a RAS molecule. Right? I didn't want to get into that detail, but what SOS does with RAS is it says, oh, you're turned off, you have a GDP with you? Give me that GDP, and I'll give you a GTP. SOS is a GDP-GTP exchange factor. Right? So what it does is it says to RAS, give me your GDP, I'll give you a GTP, and now RAS can turn on RAF, and then MEC, and then ERK. Okay? For that to happen, RAS needs to interact with SOS. And before the receptor detected the presence of ligand outside the cell, SOS and GRAB are swimming around the entire cytoplasm. Ooh, in this context, we'll see at the end of today, yes. But this cartoon that I'm showing here is way more general. So the signal that starts the whole thing could be a mitogen, so it could be something that tells cells divide. It could be a growth factor that says to these cells grow, make more proteins, make more DNA, right? It could be a migratory factor that is attracting cells to a given source. So it could be a whole number of things that is activating the RAS-ERK pathway. And the reason why these early signals can do a whole bunch of things is because the genes that get turned on down here inside of the nucleus could do a whole bunch of things themselves. Some of the genes that get turned on here in the nucleus have to do with cell cycling and self-dividing. Some of the cells in the nucleus may have to do with survival, so protecting the cells from cell death and apoptosis. Some of the genes in the nucleus might have to do with metabolism and generating more proteins and more lipids for growing. So there's a whole bunch of things that get turned on in the nucleus, and therefore a whole bunch of responses that this signal can trigger. Well, so that's what Ali was asking before. In this case, in the context of today's lecture, as we'll see later, yes. But this cartoon, as I just presented it, is very universal. And it can respond to things that have nothing to do with A beta. Yeah. It's a very general cartoon introducing the pathway that turns on RAS-ERK signaling. Okay, so now we know that when you expose cells to A beta peptides, that causes an accumulation of phospho-ERK. And they went a step further and they asked, could the incubation with A beta peptides also cause the dephosphorylation and therefore activation of GSK3? And the answer seems to be yes. So they looked at phosphorylation of GSK3 in the presence of A beta peptides, and it is reduced, even though the levels of total GSK3 are maintained. Now you have less phosphorylated GSK3, and as we just saw, phosphorylation of GSK3 is inhibitory, so having less phosphorylated GSK3 means that there is more activation of GSK3. That's the densitometry showing the decrease, statistically significant when they hit the 5 micromolar dose. And they also saw an increase in the accumulation of phosphorylated APP. In pH F1, I left that plot there, that's a phosphorylation of tau. So both phosphorylation of APP and phosphorylation of tau are increased when you incubate neurons with A beta peptides. Okay? So, in this paper so far, we've seen that incubating neurons with A beta peptides leads to ERK activation, phosphorylation of APP, and one thing that I didn't show you, but they show in the paper, is there is the induction of cyclin D1 expression. Cyclin D1 is one of these genes that get turned on by activation of the ERK pathway, and cyclin D1 happens to be associated with proliferation, with making cells divide. Okay? So this is what this paper shows. that A beta peptides trigger these three things. If you recall, in the introduction, the authors say, in a previous paper, we showed that APP led to ERK activation. So now you have these two collections of evidences, right? A beta peptides can do all those things. APP can lead to ERK activation. So how would you rearrange these observations into one hypothetical pathway? What models do we have? Who wants to share? Okay, and it stops there from phosphor. Okay, Lara. And the cycle D1 induction would be from phosphorylated APP? Yeah. See how those models, very similar, but not identical. There are some subtleties there, right? Any other ideas? Anything different than this? Or now that you see them on the screen, anything that you say, oh, I would write it differently?
Oh.
So these two models differ primarily on where we place A beta relative to phosphorylation of APP, right? One says, well, APP is what generates the AB42, and that is what triggers or activates ERK. The other one says, AB42 starts the whole thing, activates ERK, and that's what phosphorylates APP. Right? So it's basically where we place this sequence of events relative to one another. And that is precisely what these people wanted to do. See where the placement of these different things could be. Because these two models are perfectly consistent with the available evidence. Right? So you need to do some more experiments to figure out between one or the other. So let's start with this. That A beta peptides, let's say the toxic form, the 42 for argument's sake, leads to phosphorylation of APP. We saw that in the previous plot, right? So we saw that when they incubated cells with A beta peptides, there was phosphorylation of APP. That somehow leads to ERK activation, and from ERK activation to cyclin D1. So if that is your, and I'm not saying this was your model, but it was in a way the author's model, how can you test this? We add AB, of course, yeah. And then let's measure pERK. That was done already in the previous plot. Do you want to measure phosphorylated APP? Yeah. Let's measure PAPP. That was done in the plot before. Right? What experiment can you do to say, no. It's not like when you add A beta, these things happen. We've already seen it. It happens in this order. An inhibitor of what? Of ERK? Okay, and so if this model is true, what would you see when you take out ERK activity? We should see a decrease in cyclin D1, right? What will we see in terms of phosphorylation of APP? That's it. So if you want to probe this model as is, remember this, guys, because it's going to happen over and over throughout your entire careers. If this is the model that you want to support with evidence, what you do is you add the trigger, you take out the intermediate, and then you see what happens to the outcome. So to test this model as written, what we would do is, instead of taking out ERK for now, what we'll do is take out phosphorylation of APP. If we take out APP and ERK activation is still there, then that's not true. But if ERK activation goes away, then you have experimental support for the idea. So that's exactly what they did. They took cells, a cell line. These are neuroblastoma cells. So these are neurons that in this case, I think it was a rat, or it may have been a human. I forget now. These cells were in a brain cancer. So these are neurons that are dividing when they shouldn't, but they're dividing. They're not your typical neuron. These are not neurons with axons and dendrites making contacts with other neurons. These are neuroblasts, and hence the name neuroblastoma. These are neuronal cells that are in sort of some precursor state that can still divide, and they're dividing a lot, and that's how they form the brain tumor, right? So researchers many years ago took these cells out of the brain tumor, placed them in culture, and like many other cancer cell lines, this brain cancer cell line survives in vitro, and it's immortal, and it keeps growing and growing and growing. Instead of inside of a brain, now it does it in a petri dish. So these neuroblastoma cells, the B103695, do have APP in them. So in a way, this is your control with APP and phosphorylation of APP in it. So when they added A-beta to those neurons, they saw phosphorylation of ERK, even though the levels of ERK were maintained the same. And you gotta trust me here, even though in the paper they have the densitometry, there was an accumulation of cyclin D1. So when the cells do have APP, when APP is there, and again, presumably can be phosphorylated, they see that adding AB peptides checks ERK activation, checks cyclin D1 expression. But when they take cells that don't have APP, this is a cell line that doesn't have APP in it. It's B13 without APP. Now, when they add A-beta, there is no accumulation of phospho-ERK, no accumulation of actin. So effectively, what this experiment was doing was taking out AP, and as a consequence, any phosphorylation of it, out. And now you can add AB peptides, but you don't get any ERK activation, you don't get any cyclin D1 expression. So according to these experiments, you'd say, all right, well, no, that was the correct model. We're going to go with this model. But is there anything that kind of makes you uncomfortable about this experiment? Yes, and notice once again that what you see that is still present, your words,
It's a little less.
is still present, maybe a little less, but what disappears is the phosphorylation of ERK. So now, what you don't have is that active form of ERK that can go into the nucleus and, among other things, turn on cyclin D1. So ERK itself is still there, right? And I'm going to take what you said a step further. I'm going to mention this. You said ERK can be used for other things. So the fact that you don't have APP doesn't mean that you shouldn't have any ERK and ERK activation. You could activate ERK with other things. Totally agree. But without APP, ERK cannot be activated by A-beta. We can take these cells without APP, to which we added A-beta, and we did not turn on ERK, but we could add CSF, the colony stimulating factor. We could add some mitogen. We could add insulin. We could add any number of things, and ERK will be activated because it's still there. But what this experiment is saying is ERK is still there, but you cannot turn it on with A-beta if you don't have APP. Okay? Anyone else seeing a problem with this experiment?
APP is needed for A-beta. That's not a problem.
Normally, yes. So normally, you would need APP. So if the cells don't have any APP, how can you have any A-beta? You're absolutely right. What's the workaround? The scientists added it artificially. So yes, the cells are not producing any A-beta themselves, but a scientist came in with a pipetter and added A-beta to those cells. The issue with this experiment is that to test the role of phosphorylation of APP, right, that was the intermediate step in the process, you're taking out APP. That's kind of weird. To more properly test the role of phosphorylation of APP, instead of taking out the whole thing, what you should do is somehow block that phosphorylation. That's a cleaner, a more elegant experiment, right? If you suspect that phosphorylation of APP is needed for ERK activation, don't take out the whole thing, just make it less phosphorylated. And then show me again that when you add A-beta, but you no longer phosphorylate APP, you can still see or not ERK activation.